TSA agar in there, the tryptase soy agar that we've used for some of the other labs. And then it's got sheep's red blood cells in there. And that's the blood that you see. The purpose of using this agar is the sheep's red blood cells are a good nutrient for a wide range of organisms. Most things can grow on here. There's only a couple of organisms that require the sheep's red blood cells to be lysed. When they're lysed, it turns brown and we call it chocolate agar. It looks, fresh chocolate agar kind of looks like chocolate pudding, but does not have the same smell, so probably not the same taste. But what we're using in here is the red blood cells. And what you're going to be looking at is when the organisms grow on here, are they lysing the red blood cells? Yes or no? And then are they lysing them completely or partially? And that's going to give you one more hint as to what their identity is based on what enzymes they have present to do that. So for this lab, you're going to divide this up into thirds in here, and you're going to take one of the cultures, and it can be E. coli or bacillus. Either one is going to be one of your thirds. In there, you're going to take one from the sweaty armpit. So if you didn't get to use your sweaty armpit the last time, you can let a different person's sweaty armpit be it. And then you're going to take one from up in your nose. And it's got to be up fairly high. No, we're not trying to access the brain, but we're not looking for the stuff on the edge. You got to actually get in there into the nasal passageways up here to get that. So when you're done, the swabs are going to go into the biohazard box. The papers around it are not biohazard. They go into the garbage can. We try and separate that because biohazard is really expensive to dispose of. So you'll have one swab left over when you're done. Leave it in the paper container and you'll be able to use it for another lab. So nose, armpit, and one of those two that you already have. All right.